Okay, so today we're going to talk about mitosis or focus on mitosis. Previously, we looked at meiosis and we inoculated our fern plates and we worked with our pipe cleaners and hopefully we learned the fern life cycle. So we learned about alternation of generation. We learned about haplotynic and diplotynic life cycles as well. So hopefully you're all good with that by now. And today what we're going to look at is mitosis. Now mitosis is something that we do far more often than meiosis. So with humans, we produce either eggs or sperm by meiosis, which females don't complete meiosis very often at all, about 400 times total in their life. Males and humans complete it much more often, right? Because they produce a lot more sperm than females do eggs. But we produce mitosis, we produce skin cells, by the billions. We produce epithelial cells that line our guts, again, by the billions in our lifetime. So we produce lots and lots of cells where we're just cloning. The daughter cell is exactly genetically like the mother cell, okay? That's very common in our bodies. Well, some animals, like the planaria, and you can see the planaria right here in this picture. This is a flatworm, and the flatworms are just that. They're very thin and they're very flat, and they're fairly small. In this picture, they're not small, so you can come back and look at me, cameraman. All right? And so what we're going to do with our planaria is we're going to go, they'll have a small dish of them in your lab, and you'll pick out a planaria, and you're going to put it onto a microscope slide, which I think I have one right here. And you're going to take that microscope slide and you're going to place it on a small bag of ice. I don't have a bag of ice here today, so we'll have to use our imagination. And you're going to put the planaria on this slide that is on this bag of ice, okay? And then you're going to take your scalpel and you're going to cut the planaria. Now you have to wait until the planaria stops moving. The ice will cause it to slow down. And when you cut the planaria, you have some choices. So let's go ahead and look, cameraman, at our choices, okay? So it's very difficult to cut the planaria into some of these very convoluted shapes, perhaps like this one. You might just want to cut the planaria right straight in half, like you see here, just cut its head off, okay? And when you do that, you're gonna wait for your planaria to slow down. You're gonna take your scalpel and you're gonna go straight down and through the planaria, and you're going to count 1, 2, 3, 4, 5. Scoot your scalpel just a little bit. 6, 7, 8, 9, 10, and then bring your scalpel up. Okay? There'll be a little water on your slide, so the two pieces of the planaria completely separate. If you don't hold the scalpel in place long enough and keep the two pieces apart, they'll fuse right back together. As soon as if you just go cut and done, they'll fuse right back together. So you have to go in and count and make sure they're separated. Now, planaria are unusual. They're not like humans. If I cut off my arm, I don't get to grow a new arm. I won't just grow a new arm and new hand, okay? But planaria can do that. They'll grow back a head. If you make a longitudinal cut, they sometimes will grow two tails. If you're lucky, you can even get them to grow two heads. It's kind of cool. So once we've got our planaria actually here, we're going to put some spring water into another small petri dish, just like we saw. It has to be spring water. It cannot be DI water. It cannot be tap water. The planaria will die, okay? And so after we put just a little bit of spring water into the dish, we can cut the end off of a transfer pipette. So notice this is how a transfer pipette normally looks, and we're going to cut the end off so there's a wide opening, and we can then use that transfer pipette, push down, remove your planaria, and then put the planaria into the dish, get the second piece, put it into the dish, and make sure that you've labeled the little dish before, of course, you put the spring water in it. You can always label on top if you need to. And then we're going to come right down here and put it into the very bottom drawer nice and neat and shut the drawer, because this is intensive care, okay? And we're going to be very careful when we open the drawer so that any sections who have put dishes in there previously, you won't want to spill their planaria who are also in intensive care. Okay, so we want to take good care of them. And then a week from now, we should be able to see the tissue. The new growth will be gray in color, where the rest of the planaria is brown. So it's a really interesting and exciting lab, and I wish all of you a great deal of luck with allowing your planaria to divide asexually. Good luck.